And welcome. We're glad to have you to join us today for today's webinar. It's titled DYNE-251, Efficacy, Safety, and Biomarker Data from the Delivery Trial. So we're really excited to have the Dyne team here to share with us some of the data from their exon 51 skipping program. I'm going to take us through a little bit of housekeeping. I'm Liana Orlando. I'm the VP of Research at Cure Duchenne. And just to go through what we're going to do today, we're going to have the opportunity at the end of the webinar for question and answer. And so the way you could submit a question is at any time during the presentations, you can click the Q&A button at the bottom of your screen and enter your question there. If you're not seeing the Q&A button, I think if you move your mouse to the bottom of the screen, it should appear sort of in the middle of the Zoom window. We're going to do our best to answer your questions, and we'll be doing that live. But I'll also tell you that for sort of individual questions about a specific individual's mutation or situation, those are sometimes better handled after the webinar for follow-up. So any questions that we don't get to or anything that's really specific that way, don't worry if we don't do it live. That's not the end. We'll be following up with you afterwards. After today, the webinar will also be available on Cure Duchenne's website in case you'd like to watch it again or share it with others. And now I'm going to welcome to the screen Ash and Catherine from Dyne and let them introduce themselves. Thanks, guys.
Great, thank you, Liana. Hello, everyone. My name is Ash Dugar. I'm the Chief Medical Affairs Officer at Dyne Therapeutics. And first, I'd just like to thank Deborah Miller, Leanna Chelsea, and the entire Cure Duchenne organization for the opportunity today to present an extended view of our Duchenne program relative to what I presented at the Incredible Futures Conference back in May. And it's always a privilege to speak to the Duchenne community, and I want to thank you for your time and attention today. And with me today is our Head of Patient Advocacy, Catherine. Do you want to introduce yourself?
Good morning and good afternoon. I am Catherine Beaverson, and I lead our patient advocacy efforts at Dyne within the Medical Affairs organization. And just to echo Ash, it's truly a pleasure to be with the community today as we walk through the research that Dyne is conducting and updates on our DELIVER trial. Thank you for joining us today.
Great. I will gush over Catherine for a second. As many of you know, Catherine's been part of the neuromuscular advocacy community for over a decade and just has so much experience and passion that I've learned a ton from her, as well as the community, and certainly Deborah and her team. So it's really a pleasure to be here. All right, we'll get started. If we could go to the next slide, please. And we've done the intros. Next slide, please. I'd like to remind everyone that we will be making forward-looking statements today, and you can refer to our website for more information. Next slide, please. So at Dyne, it all starts with our mission, and I've presented this slide before, and it's so important because everything starts with this mission: to deliver life-transforming therapies for people living with serious muscle diseases, with Duchenne being a priority. This is woven deeply into our core values, and this inspires our employees each and every day. Next slide, please. So I'll give you a brief reminder of our FORCE platform in DYNE-251. But maybe before I even get into this, I do want to just make sure that I'm not remiss in communicating why we built the FORCE platform. There are three key challenges as to why currently available exon skipping drugs have significant limitations in terms of dystrophin restoration and functional improvement. The first is being able to get the exon skipping oligonucleotide, or the PMO in this case, historically getting enough of it into the nucleus. So really it's this nuclear localization of oligonucleotides to be able to then mobilize the cellular machinery. The next challenge has been the targeted delivery to muscle fibers, really directing it to skeletal muscle, including diaphragm, cardiac muscle, smooth muscle. So getting it really targeted with limited or no off-target effects. And then broadly distributing it in the muscles that matter for those suffering from Duchenne. So not just a little bit in those muscles, but broad and wide distribution in sufficient quantity to be able to produce dystrophin and have functional improvement. Those have been the challenges. And the FORCE platform is the solution that Dyne has developed to overcome these historical challenges. The platform is expressly designed to efficiently and reliably deliver antisense oligonucleotides like PMOs to skeletal, cardiac, and smooth muscle and target the genetic basis of disease. And it consists of 3 components, which you can see on the slide. On the top left here is the Fab, or the antigen binding fragment, or the antibody fragment. It's the top portion of this depiction of a full monoclonal antibody that's in the shape of a Y. And so the Fab is designed to bind to the transferrin receptor 1, or TFR1. The TFR1 is highly expressed across the surface of muscle cells, including the heart muscles, breathing muscles, and skeletal muscles. So the Fab targets the TFR1 and uses it to deliver the exon skipping PMO to muscle, while maintaining the ability of TFR1 to transport iron into the cells. So its natural function in our bodies is for iron transport. And we don't disrupt that iron transport. And essentially, we use this receptor to drive the PMO into the cell. And I'll talk a little bit more about that in the next slide. The second part is this, oh, sorry, if you can go back for a second. The second part is this clinically validated linker on the bottom part of the slide, which we chose because of its demonstrated safety and efficacy in third-party approved products. Because it has been used in approved products, it de-risks our program, and this linker enables the precise connection of the Fab to the exon skipping PMO. And then finally, we use an exon skipping PMO, which I think everyone here is very familiar with, which we rationally selected to target the genetic basis of disease. And this is where we leverage the modularity of this FORCE platform. And so what I mean by that is for nuclear targeting diseases such as Duchenne, we choose antisense oligo payloads, PMO in this case, to be able to target the specific mutation in the DMD gene. And DYNE-251 is our investigational medicine that uses an exon skipping PMO designed to enable skipping of exon 51 to make an internally shortened form and functional form of dystrophin. So the FORCE platform is really designed with the goal of addressing current limitations of delivery of exon skipping molecules to muscle, improving the delivery of the PMO to cardiac and skeletal muscle, including diaphragm, and then also allowing for increased time between doses because it gets in there so robustly and so efficiently. Next slide, please. So the mechanism of FORCE is different from other approaches. This is a question we often get. Again, we're essentially using the cell's natural biology to deliver our investigational medicine to muscle cells. Now, our Fab binds tightly to the transferrin 1 receptor, as I mentioned. And again, this is highly expressed across the surface of muscle cells like skeletal, cardiac, and smooth muscle. The Fab acts as a kind of key, if you will, that unlocks the TFR1 receptor and allows our PMO to enter skeletal, cardiac, and smooth muscle to target the genetic basis of disease. So in a way, we use a Trojan horse approach to drive the PMO into the cell using the TFR1 receptor without disrupting its normal function of transporting iron into the cells. So the FORCE platform harnesses the natural mechanism of TFR1 mediated delivery to transport the PMO across the cell membrane. And this is also differentiating because we're not using membrane destabilizing agents, for example, to deliver the PMO into the cell or to leave the endosome, which you can see in this cartoon here. By targeting muscle specifically by using TFR1, we also have the potential for a wide therapeutic index, resulting in less frequent dosing and chronic administration. And you'll see this reflected a little bit later when I get into the clinical program. Next slide, please. So today, Dyne is well on its way towards achieving the promise of FORCE and delivering for patients. We've delivered quite a bit of clinical data for our Duchenne program, and I'm pleased to share our latest results today. Next slide, please. Thank you. Prior to starting our DYNE-251 clinical development program, very importantly, we proactively obtained expert feedback. We engaged key opinion leaders, or KOLs, from various disciplines, including pediatric and adult neurology, cardiology, physical medicine and rehabilitation, pulmonology, and physical therapy. And importantly, we gathered this feedback from across the globe. And KOLs, both in the US and outside of the United States, with experience running early and late stage trials, have provided advice on our overall clinical development program, including our clinical study design, the patient population that we should include, the biomarker and functional endpoints, and key safety considerations. And very importantly, they provided key learnings from numerous long-term natural history studies and previous clinical trials to help inform our trial design and eventually to contextualize the results that we obtain. It's absolutely clear that KOLs believe dystrophin continues to be a key endpoint for Duchenne. We've also received advice directly from patients and caregivers, as well as global advocacy leaders such as Cure Duchenne, and from the Duchenne Community Advisory Board, comprising of advocacy leaders from several countries around the globe. They provided insights based on their living with and being the voice of the Duchenne community, as well as experience with past and current trials. And this input has been absolutely critical in ensuring a patient-centric design, and it's included, but it's not been limited to, key considerations for choosing a clinical trial, because there are so many options that it can be confusing for families. Review of and input to our informed consent and assent form, feedback on biopsies and functional outcomes, identification of the most burdensome or difficult parts of the trial, and how to ensure patients and families are supported throughout the process. And we've designed our trial and the patient support services wrapped around the trial to be able to address all of this feedback. Next slide, please. So I want to provide a brief overview of the DELIVER study design in this slide and the next slide. Importantly, it's a phase 1/2 global trial that we've designed to be potentially registrational. The trial is enrolling ambulant and non-ambulant participants with Duchenne who are ages 4 to 16 and have mutations amenable to exon 51 skipping therapy. Patients who have been previously treated with exon skipping therapy can enroll and DELIVER after a 12-week washout period. The primary endpoints are safety, tolerability, and change from baseline in dystrophin levels as measured by Western blot. Secondary endpoints include measures that you're very familiar with, like NSAA and time function tests and exon skipping. And we also have wearables to allow collection of data such as stride velocity, which in fact has been accepted as a primary endpoint by the major European regulatory authority for Duchenne trials. So we're trying to be expansive and creative in terms of the outcomes that we're collecting. For details on inclusion-exclusion criteria, you can visit our ct.gov page for all of those details. Next slide, please. So this slide goes into a little more detail on the trial design. I won't spend a ton of time on it, but as you can see here, we're continuing to step through our dose cohorts with higher and higher doses. In the multiple ascending dose placebo-controlled portion of the DELIVER trial, which is depicted here, participants are randomized to receive DYNE-251 or placebo every 4 or 8 weeks. We do take 2 muscle biopsies, one at baseline and one at 6 months when the placebo-controlled period ends. And then all participants receive DYNE-251 after the MAD portion as they enter the long-term extension. I'm really pleased to say that we fully enrolled our 40 milligram, that's the PMO dose, so 40 milligrams of PMO per kilogram dose cohort, receiving drug every 8 weeks. And we have additional 40 mg per kg cohorts enrolling now. And today I'll be reviewing the most recent efficacy data we reported, and this is at the 10 milligram per kilogram dose level. Next slide, please. So this chart shows the baseline participant characteristics by cohort. I won't go line by line, but I will say that as we've talked about, We've designed DELIVER to be potentially registrational. And to maintain the blind, we haven't broken these out by placebo and DYNE-251 treatment groups. Importantly, overall, baseline characteristics are similar across cohorts and generally representative of a population of males living with Duchenne amenable to exon 51 skipping therapy. Next slide, please. Moving on to the safety profile, which we believe is favorable to date for DYNE-251, and based on around 50 participants enrolled through the 40 mg per kg cohort, we feel very pleased with the safety and tolerability profile. We've not seen any dose-dependent increases in reported adverse events, and most adverse events were mild to moderate in intensity, and many occurred around the time of infusion. No participants have demonstrated persistent related anemia or thrombocytopenia, no kidney injury or clinically meaningful changes in electrolytes, including magnesium, and no withdrawals have occurred either. With approximately 480 doses administered in over 35 patient years of follow-up, again, we feel very comfortable with the safety profile, which seems to be favorable to date and supports us continuing to escalate doses, as I said, in the 40 milligram per kilogram cohort now. Next slide, please. So let me orient you to the slide structure here, and it's the same for the next few slides. On the very left-hand side of the slide, the gray bar represents the placebo-treated patients. The blue bars are patients dosed at the 5 milligram per kilogram dose level, and the teal is 10 milligrams per kilogram. And you can also see the change from baseline to 6 months. This particular slide shows muscle concentration. And you can see that DYNE-251 drove dose-dependent increase in PMO muscle concentration. Simply, this confirms that the drug is getting into the muscles and more gets in with higher doses. Next slide, please. We also saw a dose-dependent increase in exon skipping. So again, the gray bars are placebo, the blue bar is 5 milligrams per kilogram cohort, and the teal is 10 milligrams per kilogram. At 10 milligrams per kilogram of DYNE-251, we have nearly 2% exon skipping. So this is dose-dependent and very robust exon skipping that we're seeing here. Next slide, please. So we're particularly enthusiastic about the dystrophin data as measured by Western blot. At the 10 milligram per kilogram dose delivered every 4 weeks, DYNE-251 reached 3.2% normal dystrophin, as you can see on the left-hand side of the slide. This is a change of nearly 3% from baseline. This dystrophin analysis is unadjusted for muscle content, and we believe this is an appropriate evaluation of dystrophin. So we did the biopsies, measured dystrophin by Western blot, and we came up with, and we had 3.2% dystrophin. And these data are quite striking at a relatively low dose after only 6 months. However, we do know that others in the field have adjusted for muscle content. So for the sake of transparency, we've done that analysis as well. So on the right-hand side of the slide, you can see the muscle-adjusted dystrophin level. And we've put our calculation on this slide, which is essentially looking at our dystrophin divided by the percent muscle content to get a percent muscle-adjusted dystrophin number. When looking at it this way, DYNE-251 achieved 7.6% dystrophin at 6 months. This is a change of about 7% from baseline. And we believe this is the highest level of dystrophin reported for an exon skipping therapy for exon 51. Importantly, this is just at 10 milligrams per kilogram. And as I said before, we're currently dosing at 4 times that level at 40 milligrams per kilogram. So we're very excited to see what that yields in terms of dystrophin restoration as well as function. Next slide, please. So the 3.2% unadjusted dystrophin expression is nearing the goal of 5 to 10% that we have said for some time that we believe will drive functional benefit. And what's exciting is that as we near that target level, we see dystrophin production lead to encouraging trends across multiple functional endpoints at just 6 months. We see stabilization or improvement with 10 milligrams per kilogram of DYNE-251, while participants receiving placebo decline over the same period of time. And we see this across different measures such as NSAA, time to rise from floor, 10-meter walk run, and stride velocity. So we'll continue to evaluate functional impact at higher doses and at longer time points. And while we recognize that these are relatively small participant numbers, we are very encouraged by this data. Next slide, please. So in summary, we believe these data reinforce the opportunity for our Duchenne exon skipping franchise, starting with the differentiated safety and efficacy profile of DYNE-251. At 10 milligrams per kilogram, dosed every 4 weeks, DYNE-251 showed a relatively compelling profile with 3.2% unadjusted dystrophin, trends and improvement in functional outcomes, as I said before, like NSAA, time function test, and stride velocity, a favorable safety and tolerability profile. And again, we are currently dosing at 40 milligrams per kilogram. We do plan to provide an update on the path to registration by the end of this year. 4 exon skipping therapies have been approved in the US, leveraging the accelerated approval pathway with dystrophin as a surrogate biomarker. And we have confirmation that that pathway remains available, and we look forward to engaging global regulators further later this year. We also believe the data set from this DELIVER study illustrates the promise of the FORCE platform in developing more next-generation exon skipping therapies. And so we're excited about the opportunity to leverage FORCE to address additional mutations. We have preclinical programs targeting additional exons, including 53, 45, and 44. And there's a significant need for next-generation exon skipping therapies for these populations and more. And so we intend to pursue this. Next slide. So with that, I would like to, again, thank Cure Duchenne for the opportunity to present to you today. And thank you to the families, investigators, and sites that have made this and so many other advancements in Duchenne possible with your countless sacrifices and dedication. What you've done over the years is nothing short of extraordinary. So thank you so much. And with that, we'll take questions.
Thank you, Ash. Catherine, you're welcome to join us on screen as well. We had some questions that came in during the registration process, but again, for any of you that are listening, if you have questions you'd like to submit, you can do so by using the Q&A button, which appears at the bottom of the Zoom screen, and we'll take as many of those live as we can. So just to get started with some of the ones that came in, Ash, thank you so much for taking us through the data and the clinical trial. I'm just going to hit some of the highlights again, and you can tell me if I'm right or wrong or add more details. So the trial is still recruiting, correct?
That's correct. Yes.
And when you say that, so you're up to the 40 milligram per kilogram dose, and what would be recruiting then would be the registration cohort, as you sort of stipulated, correct?
Yeah, no, we're still going through our dose escalation phase, and so our goal, like one of our main goals for this year, is to identify our optimal dose and dose regimen. So we're really excited about the safety profile, which allows us to continue to escalate. And then we're looking at dosing every 4 weeks or every 8 weeks. So we're still, laser focused on identifying our optimal dose and dose regimen that eventually goes into the registrational cohorts.
Thank you for that clarification. And can we also maybe, just to make sure I think everybody knows, but when you're talking about this trial being potentially registrational or the registrational cohort, can you explain what that actually means?
Yeah, no, happy to. And if I'm not clear, just please let me know. And Catherine, I'm sure you can also help clarify. So we are leveraging the path that other companies have used for approval. So because dystrophin is our primary endpoint, and we know other drugs have been approved based on dystrophin because it is likely to predict clinical benefit as per the FDA guidance. We are executing a trial that is designed to be able to, empowered to show a change from baseline in change from baseline in dystrophin levels as measured by Western blot over a period of 6 months. So that endpoint, along with the safety and tolerability, will give us the data package that would be acceptable to the FDA in terms of approval based on our conversations with them and, of course, the precedent that's been set by other companies. At the same time, we also are looking at various functional measures as well to understand and learn about what our drug is doing with respect to various functional outcomes, whether it's NSAA, time function tests, wearables, quality of life, et cetera, because that's going to also help inform our conversations with regulators and also help us learn what we do with respect to our phase 3 confirmatory studies.
Thank you for that. So the idea is that the data that comes out of that would be potentially complete enough to submit to the FDA
Yes,
for
that's right.
approval.
Accelerated
Now,
approval.
that's wonderful. And since you mentioned the functional data, I see here that someone asking about the functional data beyond 6 months. So maybe you can talk about are you continuing to follow up with individuals over time? What's happening with those earlier cohorts as you're continuing to enroll the later ones?
Sure. So the way the study is set up, it is a 3-year study by design. And so we have the 6-month placebo-controlled portion, and then we have the 6-month long-term extension, and then another 2 year. The 6-month open label, and then another 96 weeks for the long-term extension. We're not just monitoring safety along that entire span of time, but we are also looking at function. So we are evaluating these same functional measures over a number of years. And to that end, we plan to, of course, report out that data, that long-term functional data, when it's appropriate. And so, yes, we're continuing to collect functional outcomes measures. The plan is absolutely to report out what we see over a long period of time. We haven't guided exactly when we're going to put that data out yet, but we are collecting it, and we will certainly share that with the community when we're ready.
Terrific.
Yeah.
And
maybe I'll pause. Sorry, I'll just pause for a second. I know, Catherine, anything to add on those two pieces on the design and the accelerated approval?
No, thank you.
Sorry, Leanna.
No, thank you. And I know you had it on one of the last slides that we should be expecting more updates maybe later and by the end of the year. Is that correct? Because we've got a lot of questions here about when will we see the 20 milligram cohort. I just asked you about the long-term functional on the ones we've already seen. And I think it's, in general, right, as the data's coming in, you'll share it as appropriate with the community.
Absolutely. I mean, I think we'll give more guidance later this year. What I would offer is for anyone who maybe heard me a few months ago, what we had said is we will provide data later in 2024. And then when this 10 milligram per kilogram data set came in the spring, we felt compelled to share this with the community. We know that there's a lot of options out there for families to think about with respect to trials and even treatment options in the US. And so we felt obligated to share that data earlier than we had anticipated. And so that's why we showed that data back in May, and actually at your Futures conference was the first conference in which we shared that data. So we were really pleased that we could put this out there, and so we'll continue our guidance that we'll put out later this year. But we do commit to being transparent about the data that we generate.
Thank you so much. And maybe we can get into the details. There's a couple, some technical questions about what the dystrophin data was looking like. We have a question about, so I think you presented percent dystrophin. Do you know about percent dystrophin expression? Oh, actually, I read the question wrong. I thought it was percent fibers, but maybe you can go back to what you were showing in those figures in terms of percent dystrophin. And
Sure.
then there's a question about what the muscle content adjusted dystrophin is showing, and maybe you could speak to why you do that kind of analyses.
Yeah. So I don't think you have to go back on the slide deck, but the dystrophin the percent dystrophin slide had sort of two pieces to it. The one piece was showing with placebo 5 and 10 milligrams per kilogram cohorts, what our dystrophin Western blot test or assay showed, which is when we took the precious biopsies from these individuals with Duchenne. We look at the level of this protein dystrophin that you all know well, and we came up with a quantification of what that looks like in the bicep, which is where the biopsy is taken. So that's 3.2%. We do this with actually a third-party lab that is running the assay, and they are incredibly credible. They are used by, they've been used widely and recognized by the FDA. And so we have a validated assay that we use that the FDA has accepted, run by a third party to give us this unadjusted dystrophin number. So at 10 milligrams per kilogram, we feel really good about 3%, 3.2%, and a 3% change from baseline, approximately 3% change from baseline, because it just shows that, yes, our drug is getting into the muscle, as I showed. It's creating exon skipping and producing dystrophin. So that's all hopefully clear. On the right-hand side of the slide is the muscle-adjusted dystrophin percentage. And that is a mathematical quantification. That's derived mathematically in terms of the adjusted percentage. The reason we do that is because we do know that in other companies, both in the exon skipping space and gene transfer space, have reported numbers where they're trying to really hone in on what is happening in the muscle tissue. So trying to eliminate the tissue that's has fat infiltrates, that's become a bit fibrotic. So really trying to say, okay, within muscle that's viable, where you would have dystrophin production, what does that dystrophin quantification look like? And so it's mathematically derived in terms of the way we do it. The formula and how we derive it is on the slide. But we really do this to be able to provide as much transparency and context around our dystrophin levels as possible. What the key opinion leaders have told us is that they appreciate seeing both numbers, one directly from the assay and the other one that's mathematically derived, so that they can have a better sense of what's happening within muscle. So it's really to provide additional context. The scientists and physicians seem to have said they like seeing both numbers, and so we wanted to put both numbers out here for the community as well. I hope that helps, Liana, to provide some.
It does. So if I have you correctly, it's one way, not just to make the data look similar to the way others are reporting, but also it sort of addresses a little bit of when you do a biopsy.
Yeah.
Each biopsy is a little variable and may have different, amounts of fibrosis or things where you wouldn't even expect there could be dystrophin, expression there. So it's sort of standardizing it that way.
Precisely.
There's a question here about the biopsies taken at baseline and 6 months, and how close are the biopsy locations? And maybe you could speak a little to that, and this variability.
So it's a good question. I don't think I have the precise answer, like how, like I assume they're saying, what's that distance looking like? What I can say is that we're taking it from each bicep, and we know from the literature and from talking to KOLs that there is variability from different muscles, so gastroc to bicep or quads or what have you. But there, one way to mitigate the variability is to go to the same muscle, and so that's what we do. So that's why we chose bicep. It also allows us to be able to go into the same muscle across the various age range that we've included in our study. So we do have individuals who are ambulant, transitional, and non-ambulant, and taking a biopsy at the level of the bicep allows us to have more uniformity and, again, decrease variability.
Thank you for that.
Mm-hmm.
I think I'm going to switch gears a little bit. There's a number of questions. I feel like the community's been talking so much about gene therapy lately.
Mm-hmm.
So there's a number of questions that I feel like are related to that kind of thinking. So I might group some here together. But one is whether or not you've tested for antibodies individuals might have to some of your force platform, like the FAB fragment. And then another question is whether or not you're seeing an immunologic response antibodies to the corrected dystrophin, because we know those are concerns with gene therapy. So,
yeah, I'll try to sort of answer this in a couple of different ways. So, just so we're all sort of baseline. So this is obviously different from gene transfer therapy, and I think the community knows that. We are not using a virus, for example, to deliver a non-native sort of protein to muscle. We are also not using a viral capsid to deliver the exon skipping drug either. We are using this, as you just said, the transferrin receptor, and we are using an antibody fragment. And as I think you're alluding to, when you use an antibody, right, you can have an immunogenic response to any antibody that's used. So we are looking at anti-drug antibodies and also anti-dystrophin antibodies. And so we don't have all of that data, yet. We're starting to accumulate, some of that data and be able to really think about if there's anything that we're seeing that we would, that would be concerning. Right now, we go through a lot of safety review committees. There's nothing that's been seen. There's been no issues with respect to immunogenic responses. We do have infusion-related reactions, which you would typically see with these kinds of constructs. So I think right now, our safety profile is quite good. I will also mention that we use a portion of the antibody. So if you remember that Y-shaped figure, we use the top sort of piece of it, so 1/3 of the antibody. What we don't have included in our antibody fragment is something called the Fc domain. And not having the Fc domain, which a full monoclonal antibody does have, helps to lower the immunogenic risk, lower the risk of complement activation, lower the risk of effector cell activation. So there's a lot of advantages to having just that portion of the antibody as opposed to the whole antibody. And then also just smaller, right? It's just smaller and gets into the cells easier. So just the mechanics of it.
Thank you so much for walking us through all of that. There's been a couple questions before during registration and now asking about plans for, and I know this will be forward thinking, looking potentially a clinical trial of evaluating your therapy for individuals who's already received gene therapy.
Yeah, that is a great question. So we what I can tell you now is we have been thinking about that for some time, about what we can do to generate evidence that will be useful to the community and physicians and payers as they think about a world in which there are both exon skipping therapies out there as well as gene therapies out there. So I don't have anything specific that I can share today, but we are thinking about it. One of the things that, I said at the outset is this is a phase 1/2 study. We really have to be very confident of our safety profile as a standalone investigational medicine, understand our optimal dose and dose regimen to be able to have the kind of, hopefully transformative effect in individuals living with Duchenne as much as possible. And then from there, we can think about how we layer on additional evidence generation, which would include thoughts around how we think about gene therapy as well. But maybe I'll pause there, and Catherine, you may have additional thoughts around that. Okay.
Terrific. So I think you mentioned it, but I know Dyne has a commitment to the Duchenne community and have already started working preclinically on other exons. So maybe you could talk about the timeline for getting to some of those, exon 53, 45, 50, 44, and whether or not those timelines would be quicker. We've got some really smart people submitting questions, whether it could be quicker than based on the data you already have for 51.
Right. Yeah, it's a great question, tough question. So what I don't have are timelines. So we are going through the preclinical development work that we need to go through for those other exons. And so we're working as fast as we can to get through the preclinical development work that we feel is critical to continue to advance those exons into the clinic. So we are moving, I think with lightning speed, as people have seen, hopefully with DYNE-251 in just a few short years from idea to proof of concept and beyond. I do think that I think to what you're alluding to, there is absolutely thinking on our part, and I know others within sponsors or academia or advocacy that are putting a lot of brain power around how to move things forward more efficiently. So for us, right, we have a platform approach, and so we should be thinking about as we are how to move things forward more efficiently, leveraging the data from the platform as we take off a 51 exon skipping payload and put on a 53 skipping payload or a 45 skipping payload. We have a great deal of information, safety data preclinically and clinically and from a manufacturing point of view on our platform, because the Fab and the linker are the same for our Duchenne therapy. So the Fab and linker stay the same, and we interchange or we exchange the payload. So we are absolutely thinking about how to move the development program forward more efficiently because we have so much information on the platform. This does require, which I believe will happen, strong collaboration with regulators, because we do feel that there should be a path to efficiently moving these forward into the clinic and into the market with a platform approach. So we're working through that, need regulators to be open-minded and open to different proposals on how to leverage a platform to move additional exons forward. So, I don't have more to offer than that, but we're really thinking through this as well as many others are.
Terrific. Catherine, I see, are there any other questions you want to address that we have listed here? There's some small things that have come in terms of, the dosing is, if it's 20 or 40 milligrams, is that weight-based? And the answer is yes, correct?
Yeah. Yes.
Yeah.
I've noticed a couple themes around the language of second generation exon skipping, and maybe Ash, you might articulate how Dyne is thinking about next generation versus just another? I think that speaks to how do we differentiate. So maybe a couple comments on the language of second generation, next generation.
Yeah. So the way that I think we think about it is, there have been a couple of second generation, if you will, exon skipping approaches. I think there are the sort of cell penetrating peptide approaches. There historically have been, a different kind of chemistry, phosphorothioates or PSOs, that other companies have, tried to move forward that haven't worked out because of safety and toxicity issues. So the way that we think about it is we've really developed this truly next generation of exon skipping therapies, specifically designed, as I said, to deliver that PMO to muscle. Not just deliver it to muscle, but have limited off-target effects, which we hopefully we continue to see as we escalate doses, creating a very wide therapeutic index, so that essentially you have a tunable, titratable, next generation exon skipping treatment for the individuals living with Duchenne. It's really tough if you have like this much margin versus this much margin. And so we believe using the body's natural cell biology, it's also giving us a very wide therapeutic index with great durability, which allows us to at least explore dosing every 8 weeks as well as 4 weeks.
Thank you. And just to build on that, I guess, to continue on, some of the line of questioning here, I think the idea is as we continue to prove on exon 51 skipping, would you see a combination of 51 skippers, or would you see this as a replacement, then thinking about combination with other approaches to treating Duchenne?
Yeah, that's a great question. That's not an easy question. I think, well, I'll preface whatever I say is not based on evidence that's been generated yet, because we don't, there's just not a ton of evidence. But one perspective is the conversation that needs to happen with your physicians in terms of what is best for your loved one, or what's best for you. Meaning, there is not just gene transfer therapy available in the U.S. now, but there's vamorolone, givinostat. There are other therapies that are on the horizon and may be approved as well in the next years. So this seems to be a world in which it will be combination therapy. What I do think an important conversation is internally truncated functional dystrophin. How should we be thinking about that versus other modalities that are producing, whether it's significant decrease in inflammation, whether it's a microdystrophin or what have you. And so the biology suggests that the closer one is to a full-length functional dystrophin, the better, based on some of the literature that exists. But it's really important to talk to those, your physicians and scientists that you rely on to be able to understand kind of what combinations make sense for where your loved one is at currently, the age, the risk profile, et cetera. And it's tough. It's a very difficult decision tree. It's a very tough set of information to digest and then make decisions on. But that's probably where I'll stop right there.
Well, you led into a very good question, which is the million-dollar question, is how does truncated dystrophin compare with microdystrophin? Are we talking apples to apples, or are we talking apples to oranges? Thanks.
Wow, Catherine, you asked me the toughest questions. This
Yeah.
is fantastic. This is completely unscripted, just so everybody knows, completely unscripted. Um, so I don't think there's a ton of evidence to be able to answer that right now. We have dug into it quite deeply, which we should, as I'm sure other academics and sponsors have, advocacy leaders as well. I think there are different avenues of thinking around this. Everything from preclinical data, for example, that's in the literature that suggests if you give exon skipping therapies and first, and then you add on a gene therapy that can probably provide a little more durability. Others will say, understand what an exon skipping therapy can do first, really try to get as much benefit out of it as possible. If it's truly a next generation, if it's producing significant amounts of dystrophin that will lead to functional benefit, because that allows you to sort of go on and off, right? I mean, you understand the kinetics, and it's something that can be, again, tunable and titratable. And then I'm sure there are also others who are talking about where gene therapy, for example, fits as well. So it's tough, but there is a need. I guess maybe Catherine, where you're going, which I agree with. There's a real need to dig into this in an evidence-based manner, understanding the role of a shortened form of dystrophin versus a microdystrophin versus a combination. And that is an evidence base that just has yet to be robustly generated. And we all have a part to play in this, from the preclinical through the clinical trials into the real world setting.
Right. Thanks for that, Ash, because I think the question is truly insightful simply for the fact of recognizing that there is a difference, right? And that can get lost in open conversations when we're talking about dystrophin readouts, that there's actually a difference in the dystrophin itself that is being produced or inserted. So thank you for that.
Yeah, no, it's a great
Yeah.
question, because it's tough. You see a 3% dystrophin number from an exon skipping and a 40% microdystrophin number from a gene transfer compound, and I don't think it's really well understood what that means in terms of, comparatively speaking, what that means.
Following up on that, I mean, this has been an excellent discussion, maybe based also on what we know preclinically or clinically from other exon skippers. What's your prediction for this restored dystrophin? Do you think it's going to keep increasing? And do you think it'll stabilize at a point, compared to the data you already have at the 6-month mark?
So for DYNE-251,
Yes.
if that's the question, yeah. So we have what I would say is pretty sophisticated PK/PD modeling. So what I mean by that is PK, pharmacokinetics, PD, pharmacodynamics. So within Dyne, we have developed and continue to iterate on this model that allows us to look at muscle concentration, skipping, and dystrophin. So based on our PK/PD model and what we're seeing in the 5 and 10 milligram per kilogram cohorts, we are producing dystrophin levels that are in line with that model. Our model predicts that at higher doses, we'll get to 5 to 10% unadjusted dystrophin. So when you ask me what we expect as we go to higher doses, I do expect increasing levels of dystrophin, of percent dystrophin, with the 20 and 40 milligram per kilogram doses. But that's also just a prediction based on the PK/PD model. So I do not see dystrophin restoration, saturation, or sort of leveling off in sight for now.
Terrific.
Does that answer your question, Liana?
Yeah, I was also wondering if you think you might even get higher than that if beyond 6 months, right? Do you expect there might be accumulation that happens on a longer time frame as well, and whether your models speak to that.
Yeah. So the answer is yeah, of course. So great question. Our model predicts, past 6 months, of course, goes out for, a couple of years. And the literature suggests that, dystrophin sort of reaches steady state, maybe somewhere between 100 and 136 days, or, something around there. So, you want to be able to look at a model that goes 1.5 to 2 years out, which is what we do. And so based on, again, our modeling, but also based on what has been seen with compounds like eteplirsen,
Exactly.
which shows accumulation up to even 180 weeks. Dystrophin does accumulate within the cells, within the muscle. So our model takes into account a longer time period and accumulation of dystrophin. So we do expect that if our prediction is right, we would get to higher levels than 5 to 10% over a period of 1.5 or 2 years. So it's a great question. And then the only other thing I'll add is function, right? At the end of the day, we can talk all we want about dystrophin and 5% or 10% or 15% microdystrophin, whatever it is, but it really is at the end of the day, is there some functional benefit to the families, to the patients? Are we seeing changes in muscle, in mobility, in upper limb function? Are we seeing stabilization or slower progression in any of those parameters? Are we seeing stabilization of cardiac decline? Are we showing stabilization of pulmonary changes? So it's really being able to show that we are significantly decreasing decline, hopefully hopefully stabilizing, and then it would be great to be able to show that there's a little bit of gain in function as well, or a lot of gain in function. So we can talk a lot about dystrophin, but we also talk all the time at Dyne, and I'm sure other companies, it's really function. Need to show that there is some functional benefit as we get to higher doses with higher dystrophin levels over time.
Thank you. Well, I'm keeping an eye on the time, and I
Yes.
think we're coming up to the close of our hour. I just want to acknowledge that there's a couple of questions in Spanish, and nosotros no hablamos español muy bien, so we will have our partners help with addressing those questions in Spanish after the webinar. And I just want to first thank both you, Ash and Catherine, for making time today to come and speak to the community, to be so forthcoming with all of the questions. We really appreciate it.
Our pleasure.
Always a pleasure. Thank you.
And for all of you still listening, thank you so much for taking time out of your day. I hope this was helpful to all of you. There's going to be a post-webinar email, which will be sent tomorrow, should you have any other questions. And we'll get in touch with all of the attendees to let you know where you can watch the webinar on our website. And you can share that with others if you think they might be interested as well. And with that, I think we can all wave goodbye and say thanks again. Hope you have a wonderful day.
Thanks for joining. Bye-bye.
Thank you.